Good morning, everybody. I thought I would do a quick review video. I know that we went over the questions and the slides in class, but I wanted to give any of our students who may have been absent and any students just a second review, another opportunity to look at some of the slides. Plus, I wanted to take some time to go into depth with some of the slides that we didn't get a chance to do in class. So let's go ahead and take a look. All right, these are the 5 standards that are covered in our genetics unit. So if we take a close look at these skills, we're going to be able to see that these 5 standards are things that we are going to be asked in a variety of different ways to demonstrate our ability to do. So first of all, we need to think about what do proteins do? And basically the big idea of this slide is that structure is related to function. The way something is structured, the way that it is physically designed, dictates its function. And the ability to function is determined by the structure. So if something happens to that structure, then the function is not going to be what it should be, right? If we think, for example, if we were to take a bridge, a really well-designed bridge, and we take a big chunk out of the center of it, right? We change the structure, it's no longer going to be able to perform the function it was designed for, right? There's gonna be some problems. So when you think about the functions of proteins in the body, proteins are three-dimensional shapes made of these things called amino acids. So there's about 21 amino acids that make up all of the thousands of different proteins in our bodies. Just like letters make words, amino acids make proteins. Just like words have different meanings and different purposes, well, so do the different proteins. So our proteins have different functions. I'm going to start with the picture of the muscle in the top left corner. So actin and myosin, which are long strand proteins in our muscle cells, their job is to contract or to pull the muscles close, which creates movement. So a function of proteins in our body is contraction, or we say movement. Another thing is that all the hormones that are in our body, those are proteins as well. Things like insulin, which is created by our pancreas to break down sugars, carbohydrates, and sugars. Estrogen and progesterone and testosterone, those are all hormones as well. Whenever we get scared at something that we see and we get scared, that's a hormone, right? It's a hormone that triggers our body. Maybe it triggers our heart rate to increase. Dopamine, serotonin, those feel-good chemicals in our brain when we're happy or when we've done something exciting, those are hormones as well. The third function is protection. Our white blood cells produce proteins called antibodies. And those antibodies attach themselves to viruses and kind of break down and can attach to and help break down and signal to the white blood cells to attack viruses and bacteria and other foreign things in our body. The next thing that proteins can do is transportation. One best example of that we had earlier in a lesson this year is hemoglobin. So our red blood cells are red because there is this protein on our red blood cells called hemoglobin. And hemoglobin's job is when our red blood cells go into our lungs, oxygen from the air that we breathe attaches to the hemoglobin and gets transported to all of the cells of our body. So hemoglobin does a really good job of transporting oxygen to the cells that need it. Another function are enzymes. There are thousands of enzymes in our body, in our cells, doing lots of different things. When we talk about the process of meiosis or mitosis, right, cells making new cells, all of those jobs that start things and stop things and make things happen, those are all done by enzymes. So there's enzymes in our cells, and then our body also produces enzymes, for example, digestive enzymes, which have the job, enzymes in our saliva, which break down starches, or lactase. For some of us that produce lactase, that breaks down lactose, which is milk sugar, or all sorts of different pepsin and all sorts of different enzymes in our digestive system that break down food. Another function of proteins is storage. The example they give here is ferritin. So our hemoglobin has iron in it and needs iron. Sometimes we might eat a meal that has lots of iron, and sometimes we might not. And so if we look at human history, right, there was times when animals were harvested or would go hunting, but then there might be stretches where you're eating mostly stored fruits and vegetables. And so our body has a mechanism for storing that iron when we get it. And that's in ferritin. Another purpose of protein is structure. So your hair is a really great example of that because they use the example of spider silk. But on the human body, hair is a really great example of a protein. Hair is not a cell, right? Hair is this really long stranded protein that your hair follicles are constantly making. Same thing with your fingernails. Your fingernails are keratin, which is produced by the cells in your nail bed. So our fingernails, our toenails, those are all long proteins that are just a structural protein. The last function that we have written here, though there's some others, is receptor. So we have receptors on all of our different cells and in different parts of our body to receive information. So for example, in our nose, up in our nose, we have our smell receptors, right? If mom or dad or we go to a bakery and there's chocolate chip cookies being baked and we smell that, right? It's molecules from the cookie are entering onto the protein receptors in our up in our nose, right? And it's receiving those molecules and telling our body, that's delicious. So let's talk about the process of protein synthesis. We've got the process of protein synthesis, right? Or sometimes in our text, they called it gene expression. So it's the process of taking our DNA, which is just the genetic code that's in our nucleus, and turning sections of that into protein, so expressing or showing the physical structure made from the DNA. So remember some things about DNA. DNA is double-stranded, and it has 4 bases, 4 nitrogen bases, and the sequence of those bases tell our cells which amino acids to lay down, and that tells which proteins are being formed. There are sections of DNA that are non-coding sections. That makes up about 98% of our DNA. About 2% of our DNA are coding sections. So those are the sections of DNA that make the physical structures that make up our body. So how does this happen? DNA is always inside the nucleus, but proteins are formed in the ribosomes. So there's kind of a complex process to get from DNA to there. So let's break it down. First step is transcription. In transcription, enzymes split open the DNA, and other enzymes read the DNA sequence. They read the DNA sequence and they create another type of strand called mRNA, messenger RNA. Now, that messenger RNA is a shorter strand than DNA, and it's the strand that covers that gene that's going to make that protein. Now, that messenger RNA, M for messenger, M for moving, that messenger RNA is going to move. It's going to leave the nucleus. It's going to take the genetic code from the DNA. It's going to move it out of the nucleus and into the ribosomes. That's transcription. In translation, think of transcription as like transcribing, writing something down and getting the information somewhere else. In translation, we're going to take that genetic information and we're going to turn it into something completely different, right? Translating, for example, if I translate something from English to Spanish, I'm going from one language to another, something completely new. In translation, the mRNA enters the ribosome, which itself is a protein. It's going to enter the ribosome. The ribosome is going to read the messenger RNA and transfer RNAs. It's going to take amino acids from all that are hanging out inside the cell from the food that we eat. It's going to take the amino acids. It's going to take them using tRNA, and it's going to bring it to the ribosome and create an amino acid chain. That amino acid chain is the basis for the protein. And then when the amino acid changes, the chain comes out and it's going to fold on itself to make your overall protein. So, sum it up super quick. Transcription. DNA gets read by enzymes to make mRNA, and the mRNA travels to the ribosomes. In the ribosomes, the mRNA gets read by the ribosomes, Amino acids from all through the cell get brought and into the ribosome, and the amino acid chain is made based on the sequence of the mRNA. So the first step of transcription is to take the DNA sequence and turn it into RNA. Again, A's and T's pair together, C's and G's pair together. Now, with mRNA, however, they don't have T's, so they have to use U's. They have to use a different base called a U. Same as sometimes you might be baking a cake, and instead of using butter, you have to use margarine instead because you don't have any butter. Almost the same, but not exactly the same, slightly different in structure. So what would the sequence be for this mRNA? The mRNA sequence to this would be A, G, C, A, G. Now there's an A there. The mRNA would be a U, a C with the G, and then an A. So when you're looking at these slides, there's this video, and I really, you should definitely take a look at it and look at the different, what are mutations, what are the types of mutations, and what are the impacts of those mutations. There are 3 types of mutations that we think of. There are insertion mutations, deletion mutations, and substitution. And they really are exactly what they sound like. In an insertion mutation, an extra DNA base gets added. And when that happens, the whole sequence has to shift. Imagine there's a line of 20 people, and somebody cuts the line 5 people back. The first 5 people, right, they're still first, second, third, fourth, and fifth. But everybody from the person that cut, now that new person is in the 6th person in line, and everybody else has moved down a position. Now, if the teacher comes along and counts you off and groups you up in groups of 3, now the groups of 3 will be completely different from person number 6 all the way down. The other big type of mutation is deletion mutation. So imagine, again, those 20 people are standing in line and somebody says, oh, man, I don't want to be here. I'm out. And they leave. Everybody moves up in line. Imagine the teacher groups you up into groups of 3. Same thing. Now, from where that person left all the way down, all those groups of 3 are going to be different. And so if we looked at the codon wheel, right, those may all produce a different amino acid, way different protein. The third type of mutation can have a large impact, but sometimes it may not have any impact at all. And that's a substitution mutation. That's where in the sequence, one letter might be taken out and then another one put in its place. So nothing around it is impacted, just that one base. That base might impact an amino acid in that group or might not. And that takes us to our next question. Does a change in DNA always result in a change in protein structure? Nope. Not always. In order for there to be a change, there has to be a change that causes a change in a amino acid. So for example, if our RNA sequence is UCU, but there's a substitution mutation that makes it a UCC, well, that still codes for serine. There's no change in the amino acid, and that means there's no change in the protein. So for there to be a change in the protein, the mutation has to be this situation. For there to be a change, it has to be in the coding region of the DNA, and it has to change the amino acid sequence. Now that moves on to how do these genetic sequences move into offspring? Remember that we all have 23 pairs for a total of 46 chromosomes. For each pair, we have a matching pair. One that we inherited from mom, one from dad. If we have more of any of those chromosomes, then we have a chromosomal difference, and that results in some different symptoms or physical expressions. So let's break this down. In order to form a fetus or a fertilized egg that has the potential to grow into an individual organism, an egg from a mother and a sperm from a father come together. The egg and sperm each have 1/2 of a set of chromosomes. The egg and the sperm together give the baby the full or the whole set of chromosomes, right? So the fertilized egg ends up with 23 pairs, right? Imagine you get 23 left socks from mom and 23 right socks from dad, and then you end up with 23 full pairs of socks, right? If mom accidentally gave you 23 individual socks, but actually gave you an extra pair of something, you can have a little extra chromosomes, you're not going to have the right number of sets, or dad in the sperm as well. So overall, half the DNA comes from one parent and half the DNA comes from the other. When we look at this process of how does that happen, how do we get from a cell down to a gamete cell, like a sperm or egg, that's different than the process of, let's say, our skin cells, right? We're losing skin cells all the time. Our liver cells are constantly replacing themselves, kidney cells, blood cells, even bone cells. Those cells replicate the way that we talked about when we talked about the bacteria. They start with a cell, they double everything, including their DNA, and then they divide into two exact copies of each other. In mitosis, for your skin cells to make new skin cells, it's really important that they make identical copies. The goal of meiosis is not the same. The goal of meiosis is a strategy for sexually reproducing organisms to survive. If we think about the juncos and all their genetic diversity, and we think of mutations that cause traits, and then this new unique trait that allows the species to survive better, or a combination of traits that allows a species to survive, that's good for a species. And so the process that happens in sexually reproducing organisms is meiosis. We have a parent cell. So let's walk through meiosis. A parent cell doubles its DNA. And in that first step, it doubles the DNA, but then something different happens in meiosis than happens in mitosis. If we look at this process, there's an exact copy of the DNA over here. Same thing, copies of DNA, but the DNA mixes up. It's called crossing over. And genes get mixed up to create new varieties, right? It's like putting a bunch of things in a bag and shaking the bag around to mix things up really well so that you're getting a random pull out, a random chance at getting different traits. And so this is kind of what happens in crossing over. Genes between the sets, the pairs of chromosomes, they mix and match in all kinds of unique combinations, millions of combinations. They divide into two daughter cells that still have a full set of DNA, but now their DNA is unique and different. One daughter cell is different than the other daughter cell. Some genes have been mixed and matched. In meiosis, there's an extra process where now those two daughter cells divide again. And when they divide again, they divide into four daughter cells that each only have one of each pair. They have 23, in humans, they have 23 chromosomes in each. One of each of the 23 pairs. Those are our gametes. Those are our egg cells, or those are our sperm cells. So if we look at mitosis and meiosis, there are some very specific differences and some similarities. One, they both start with a parent cell. Two, the DNA doubles. And three, the new resulting cell that gets ready to separate has two sets of pairs of 23. So we have pairs of 23, so 46, and there's two sets of those. So we're looking at 92 chromosomes in there. This is where things get different. In meiosis, the genes cross over. In mitosis, they do not. Everything stays the same so that we get identical cells. In mitosis, the cells split into two daughter cells with full sets of DNA and that are identical to each other. In meiosis, they split into two daughter cells that have full sets of DNA but are not identical to each other. Mitosis 2 then happens, resulting in 4 gamete cells that have half the amount of DNA. So overall, mitosis' job is to produce body cells. Their job is to repair and replace tissue, any tissue, muscle tissue, skin tissue, heart tissue, bone tissue, whatever tissue the cell comes from. It also is the growth of the body, right? We are not the same size we were when we were born. It's because our cells and our organs and in our body have reproduced to produce more of those cells and we get bigger. If we're talking about other organisms like bacteria, that's called asexual reproduction. Right? There's no exchange of genetic material. It's kind of like a cloning process. And the same number, the chromosomes of the daughter cells, the ending cells, have the same number of chromosomes at the beginning. Meiosis produces 4 sex cells, and it's all about creating variety or variation through recombination, through recombining those genes. It reduces the number of chromosomes in the resulting cells by half, so we end up with half the number of chromosomes. We talked about inheriting traits. So in this case, we would say the gene color for fur has 2 alleles. The dominant allele F codes for gray fur, and the recessive allele little f codes for black fur. The dog is heterozygous. If we're talking about heterozygous, that means it has one of F. So that's big F, little f. The male dog is homozygous recessive. Recessive alleles are alleles that hide, and there's a video that I've already posted for that, so go check that out if you haven't already. So homozygous means 2 matching letters. And here's the trick. If something's recessive, if an organism has a recessive trait, it's always homozygous recessive. It always has 2 of the little letters. You cannot have a recessive trait and have a dominant version of that gene, because then you'd have the dominant trait. So anytime you see recessive, right away you should think 2 lowercase letters. That's going to be helpful in the next one. It says figure out the percentage or ratio of possible phenotypes and genotypes using the Punnett square. So a female dog is heterozygous. By the way, another trick, heterozygous means that ever see heterozygous means they have the dominant trait, because it means they have at least one dominant allele, one dominant version of the gene. So the female dog is heterozygous. That means that she's gray, by the way. And she's going to have, so over here, we're going to put big F, little f for mom. And for male, we're going to put little f, little f. And we're going to fill out the Punnett square. So we're going to have what would be big F, little f here, big F, little f here. So 2 of the 4 boxes. And this one would be little f from mom, little f from dad, and same thing here. So for this combination, these 2 boxes are telling us the possibility of 2 out of the 4 boxes, or 2 out of 4 chance of the offspring being gray. There's a 2 out of 4 or 50% chance of the offspring being black.